have our last speaker, one of my heroes, Eric Verdin. Please join us on stage.
Morning, everyone. Let me start by thanking Martin and Alex, not only for the invitation to speak, but also for, I think what we all agree is a really amazing meeting, especially after being sort of all segregated for a couple of years to get back together and to celebrate science. I think it's really special. So thank you for putting this meeting together. I want to start by apologizing about if you're looking at the program, you will see I had a completely different title. I was supposed to talk about an epigenetic clock, but we're not quite ready. So you'll have to come to the next meeting. I'm speaking again in Berlin in a couple of months, and I will be discussing it at that time. I decided to focus instead on a long-standing area of interest in the lab, which is the biology of the ketone body ß-HYDROXYBUTYRATE. And just a couple of disclosures. I'm a founder in a number of companies that are commercializing ketone body esters. I'll be discussing those a little bit at the end. So my lab has been interested at the interface of biology and biochemistry. And one of the most robust way that one can induce increase in lifespan and health span in many animal species is calorie restriction. And so you have the intervention, calorie restriction, you have an increase in health span and lifespan manifested by longer life, but also a decrease in the disease associated with aging, and there's a black box in between. And we've been focusing and proposing the model that this black box is filled by variation in the relative abundance of a number of metabolites, and I focus on a few here. There are obviously many others that include NAD, acetyl-CoA, S-adenylyl methionine, alpha-ketoglutarate, and ß-HYDROXYBUTYRATE, the ketone body. One of the remarkable findings that has emerged in the last few years, 10 years, is the idea that many of these metabolites are not only playing key role in nutrition, in intermediary metabolism, but they are also signaling to epigenetic regulators. And you can see many of these pairs that have been identified, NAD interacting with sirtuins, acetyl-CoA regulating the activity of acetyltransferases, and in particular for the presentation today, the idea that ß-HYDROXYBUTYRATE also functions as an inhibitor of histone deacetylases. And so this is the area that I'm going to be focusing on. So what are ketone bodies? They are first and foremost nutrients. And this is really important to remember. This is how biochemists and diabetologists and endocrinologists think about them. So when you are fasting, the primary source of energy becomes the adipose tissue at the bottom. These adipose tissue released triglycerides and fatty acids circulate, end up in the liver, and a fraction of them, of these fatty acids, get converted into these ketone bodies, which are then distributed to the whole body as a source of energy, in particularly the heart and the brain. Now there are, and remarkably, when you look at, this is something that's not often appreciated, is the amount of ß-HYDROXYBUTYRATE that's generated during a state of ketosis. So if you are completely fasting, your liver produces about 150 grams a day of these ketone bodies. And you can see plasma levels start accumulating within 18 hours of a complete fast. By that time, they can reach close to 1 millimolar. If you're fasting for more days, you can see that you can have plasma ß-HYDROXYBUTYRATE level that reach up to 6 millimolar. And of course, there are pathogenic conditions such as diabetic ketoacidosis where these levels can go up to 30 millimolar. So this is a nutrient that circulates in the plasma with relatively high concentrations. Now, there are other ways in which you can induce a state of ketosis that is not fasting, and I've listed them here. Exercise is a potent inducer, especially with a mild fast, but also some forms of dietary restriction. It turns out that calorie restriction is associated in most cases with a mild level of ketosis. And finally, the way that has been used in the popular press the most is the idea of the ketogenic diet. which can be induced simply by restricting carbohydrates, but to an extreme degree. Just to give you an idea, in most humans, if you want to enter a state of ketosis, you have to restrict it to less than 15 to 20 grams of carbohydrate. That would mean an apple a day and no more carbohydrates. If you do this within 2 or 3 days, you will enter ketosis and with plasma levels of 1 to 2 millimolar. So, how we got into this is somewhat sort of serendipitous, as some of the most fun science can be. My lab cloned the families of histone deacetylases. We cloned HDAC3 through HDAC7 back in the early 90s. And I was struck at the time by the fact that one of the known HDAC inhibitors, butyrate, which is a bacterial product that is actually present in our intestine, was quite similar to ß-HYDROXYBUTYRATE, which is the major ketone body. And so we decided to test the kind of simple hypothesis: could ß-HYDROXYBUTYRATE actually function as an HDAC inhibitor? And indeed it does, and this is a dose response that shows you BHB at the bottom on the x-axis, the concentration, and on the y-axis, the relative activity of different HDACs. And you can see that many of them actually are suppressed by concentration of BHB between 1 and 3 millimolar. You get 50% inhibition, which we know is likely to be significant. In this initial publication, where we reported this suppression of HDAC by ß-HYDROXYBUTYRATE, we also identified a number of targets. And this is where I think we got quite excited, because these targets, one of them, the most prominent one, was FOXO3A, which we know is really associated with aging, especially in humans. And this suggested that there might be some kind of biology, some biology of aging linked to the ketogenic diet and ß-HYDROXYBUTYRATE. Now, the story got very quick. So these data suggested that ß-HYDROXYBUTYRATE is not only a nutrient, which we know is consumed, but also a signaling molecule. And the story then got very quickly much more complicated, and I've listed here all of the other signaling functions that have been identified for ß-HYDROXYBUTYRATE. And in some way, from a mechanistic standpoint, in the future, it's going to be very difficult to untangle all of these signaling properties from another. That being said, I want to review them very briefly. Vishva Dixit is actually, who's actually in the audience today, reported that BHB also inhibits the inflammasome, by a mechanism by which it could exert some profound anti-inflammatory effect. Others have reported that BHB can actually signal to some cell surface proteins, HCAR2 and FFR3. We've reported, as I just mentioned, that it can function as an epigenetic regulator by modifying BHB. And others finally have reported that BHB itself can be a modification that can target histones. And this is shown on the left. So, and we are still working on trying to entangle all of these variations. There are interesting forms, and disentangling this not only one from another, but from the nutrient aspect of BHB. Now, one aspect that's interesting is we have non-metabolizable versions of BHB. The L version cannot be metabolized, but we've shown it still signals. And so that's one aspect. There are other ways in which we can explore this. So a number of years ago, we asked a very simple question. Is a ketogenic diet, which, by the way, has been gaining a lot of traction in the popular press and in the lay press, so we asked, can it be protective against aging? And so we did a whole lifespan study, which actually showed that indeed mice placed on a ketogenic diet show a reduced midlife mortality. You can see here we have no increase in total life expectancy under the experimental conditions where we did this experiment, but you can clearly see, especially early in life, a decrease in mortality. And this is quantified here, where we've, what I'm showing you on the y-axis is a chi-square test of day-by-day difference in survival. You can see a really early effect of survival. Now, one of the complications of the study that we did here is the fact that These mice never saw a carbohydrate in their entire life. Actually, that's not totally true. They were alternating between a ketogenic and a normal diet. The reason why our initial experiments, we fed them only the fat and the protein, and they ate so much they became absolutely enormous. So we had to stop the experiment. And so we alternated 2 weeks on, 2 weeks off. And despite this, we still saw this strong effect. Separately from our study, another group, John Ramsey and Jose Alberto Lopez Dominguez at UC Davis, did the same experiment. They put mice on a ketogenic diet, but they fed them a certain amount, a fixed amount of food, which means the mice actually did not gain weight. So they were not on an ad-lib diet. And they showed an even stronger and more robust effect on life expectancy under these conditions. So perhaps what generated even more excitement in the lab was the idea that we saw some remarkable phenotypes in terms of CNS function. And so the next question we ask is the ketogenic diet protective against some of the chronic disease of aging or manifestations thereof? And because this data is published as well, I want to review them very briefly. We've conducted a number of tests that assess CNS function. For example, elevated plus maze exploration. You can see on the light blue shows you a basal. The dark blue shows you the aged mice, where there's a loss of function, and mice on a ketogenic diet essentially do not show this loss. The same thing for interactive place avoidance. You can see actually in this case, the mice on a ketogenic diet behave even stronger than mice, than even in younger mice. Other tests that we've conducted, just to give you an idea, a so-called novel object recognition in old age. Again, you can see an increase in a ketogenic diet, and particularly with a familiar object. I'll skip this. So all of this data has been published. I want to focus today on some additional studies that we've been conducting, looking more specifically at a model disease for Alzheimer's disease. And this collaboration was initiated based on this publication from my colleagues at the Gladstone Institute, Jorge Palop and Leonard Mookie. They reported that in a variety of Alzheimer's models, there is a defect in inhibitory interneurons. And this seems to be playing a critical pathogenic role in Alzheimer's. And so just to give you a bit of background on what this means, you're seeing here the cortical neurons that are here, normally under the controlling or regulatory influence of so-called inhibitory interneurons. Then these interneurons depend on a unique channel here, SCN1A. Under conditions of Alzheimer models, the APP mouse, or under other conditions, this so-called Dravet syndrome, which is actually a disease of childhood associated with childhood epilepsy. These patients actually carry a mutation in this channel here, SCN1A. The inhibitory activity of the interneuron is lost, and there's epileptiform spikes generated by the cortex. This is associated with epilepsy and disturbed cognition. So there was another thing that was quite interesting about this is the fact that the treatment for Dravet in humans, actually in children, the best treatment is ketogenic diet for reasons that are not entirely clear. Another thing that's interesting is that one of the other known drugs that is treating that conditions, valproic acid, has also been reported to be an HDAC inhibitor. And so all of these convergent data suggested to us that maybe placing these model of Alzheimer's on a ketogenic diet could actually be an interesting way to explore therapeutically. And so again, in collaboration with Leonard Mookie and Jorge Palop at the Gladstone, we obtained these mouse HAPP-J20, which is a well-established model for Alzheimer's disease. They express a mutant form of human APP, which bears both a Swedish and an Indiana mutation. And so this an abnormal form of APP is expressed under the control of a neuron-specific promoter, PDGF-beta, and this results in several key aspects of Alzheimer's disease in these mice. They show deficit in learning and memory. They have behavioral abnormalities, synaptic alteration, amyloid plaques, gliosis, and spontaneous epileptiform activity, which in collaboration with Jorge we could measure. So we basically did the experiment that that I'm leading you to. We placed these mice on a ketogenic diet, 10% protein and 90% fat. The control diet was actually isocaloric in terms of proteins and just simply added carbohydrates. And we were able to show, this is the data here, but this is its quantification, that normally the ketogenic diet was able to to very significantly suppress the increase in epileptiform activity that's shown here. And interestingly, sort of an acute fasting didn't. So, and under these acute fasting conditions, you don't see ketosis. So really suggesting the idea that even with a brief period of ketogenic diet, this is just a couple of, I think, 3 weeks here, you could actually revert a key manifestation of this abnormal mouse. Not only did this, we were able to show that a ketogenic diet had a remarkable effect on memory in these mice. Again, we used a training open field, and you can see first showing you that these mice were indeed in ketosis, in this case about 800 micromolar. Using a number of measurements that are associated with the training open field without getting into the details, if you look at the normal mouse, you compare it to the APP mouse, you see an increase in total movement, which is abnormal. This is suppressed by the ketogenic diet. Same thing with center movement, a dramatic increase here, completely suppressed, same thing. So by every manifestation, every measurement that we could make of this open field test, we saw a correction of the defect. Remarkably also, these mice have a very short lifespan, especially in the males. And you can see normally all these males are actually the mutations, the transgene leads to early lethality within 180 days, and we had a pretty strong increase in life expectancy in mice on a ketogenic diet. This was much less evident in the female mice. Now, this is very nice, so that would suggest that maybe a ketogenic diet could have some applications in Alzheimer's, and if you go again in the popular press, which we should not base our science on, but there is evidence of patients actually making significant recoveries in terms of cognitive function while being switched on a ketogenic diet. So but we also know that these ketogenic diets are extremely hard for people to stay on. Just imagine restricting your carbohydrate to less than 15 grams a day. So we've been very interested in trying to determine whether we can recapitulate the effect of the ketogenic diet with exogenous ketones alone. Now, I'm going to show you here, this is the structure of ß-HYDROXYBUTYRATE. You can see here, you can today go buy on Amazon ketone salts, and you can eat them. I just found out recently that the major seller of these ketone body salts sells for about $100 million a year of these products, which, by the way, do not work. The reason they do not work is if you ingest ketone bodies, they get rapidly absorbed. You will induce ketosis within 30 minutes. 30 minutes later, it's all gone because they are extremely rapidly metabolized. And so the field has been struggling with this and trying to find alternative ways of or delivering these ketone bodies. And the way we and others, this is work that was pioneered by Dr. Veach at NIH back a number of years ago. We've been synthesizing, he was the one who pioneered the idea of making ketone body esters, derivatizing these ketone salts to increase their persistence and their delivery. I should say also one of the other problems, because this is an acid, it needs to be complex with a salt, and administering the ketone salts is associated with a significant burden of either magnesium citrate, calcium, or sodium. And so practically, I would say the ketone salts themselves are not a solution to this problem, especially long term. So we came up with a different way of making ketone body esters, which was to couple ß-HYDROXYBUTYRATE, shown here, with a short-chain fatty acid. Now, short-chain fatty acids, you might remember from the very beginning of my presentations, are able to go to the liver and to make ketone bodies themselves. So the idea behind these molecules is that you have, once the ester, and we have different length of chains of fatty acids, these will be absorbed, will be deesterified, which will generate the ketone body itself, which induces ketosis immediately, and a fatty acid which goes to the liver and has sort of a longer-term effect in terms of inducing ketosis. So think of them as a prodrug, and that also delivers the drug. And so this actually works remarkably well. What I'm showing you here is the level of plasma ß-HYDROXYBUTYRATE, shown here, hours after injection. You can see at the highest dose, at least one of these generates a stable state of ketosis. And with the salts, this would have all been done within an hour. We have conducted a lot of work with these. The companies that I discussed at the beginning actually are commercializing this and testing these in clinical trials for a number of conditions. But I want to show you also some very preliminary data showing that these by themselves actually were able to suppress the spikes that we started with. And so we think, and we have preliminary evidence that these actually also revert and exert positive effect, not only in terms of brain function, but in a variety of other experimental systems. And so this obviously is work that's ongoing to be hopefully confirmed in the future. I want to finish here and acknowledge the people who have participated to this work. John Newman has been a postdoc in my lab and now is a faculty at the Buck. Tadahiro Shimatsu was the early person who generated a lot of this HDAC work. Matt Hershey also, who is now actually a faculty at Duke, and others. Thank you very much.
Thank you so much, Eric. That was really fantastic. We have a question.
Great talk. I have a question. So kids with the disease tuberous sclerosis, so they use...
Which disease?
TSC, tuberous sclerosis complex,
yes.
Right? So they use ketogenic diet as a therapy. So in some kids they use it, some kids don't. So the question is, so the kids on the ketogenic diet, they stay for a very long time. So do they have any benefits in terms of the aging phenotypes later in life? And second, whether exogenous ketones can be used as an alternative therapy for these kids instead of ketogenic diet, because it's so hard for them to stay on ketogenic diet. Thank you.
Well, yeah, tuberous sclerosis is a really interesting aspect. I don't have the data, and I'm not familiar with the paper you're referring to, but I would predict that most of the beneficial effect they see in those patients on a ketogenic diet is not due to ß-HYDROXYBUTYRATE, it's due to the carbohydrate restriction and low insulin signaling, since we know, these tuberous sclerosis have very high mTOR activity. So one way you might be able to mitigate this is by simply restricting carbohydrates. I cannot comment further on this, but I can see many ways in which, I know people are looking at rapalogs actually in these kids as well as a way to suppress some of the manifestations. But in terms of how this works, again, and this is something I tried to convey during the discussion, these are pleioform molecules that have multiple effects. At the end of the day, if they are effective, I don't really care how they work, but I think there's a lot of work trying to really dissect all of these different mechanisms.
Thanks, great talk. How did you key in on ketones in the brain? What was kind of the initial connection there?
Oh, why did we focus on this aspect versus the rest? I should say we're focusing on many other aspects. It was really the strongest phenotype that we got in terms of mice on a ketogenic diet. I mean, clearly we screened for everything. But the memory aspect, the fact that, for example, mice on a ketogenic diet actually have better memory than young mice is still really striking to me. And that was the reason.
Thank you very much for the valuable lecture. As a part of our intervention for obesity and insulin resistance, I'm a nutritionist. Okay, we recommend a ketogenic diet for this, and especially for the epileptic children. And we have seen a dramatic effect. Some of them completely relieved. Most of them, we decreased the dose of dipakine and levetiracetam to a very low dose. The point that I wanted to add is that when we add too much, protein within the ketogenic diet, because 20% that it contains a glutamic acid or glutamate, which is exactly inhibit the effect of ß-HYDROXYBUTYRATE. Because as you know, GABA, which is a very good thing, which is like a similar component of butyrate or ß-HYDROXYBUTYRATE. So when we decrease the protein also into 10 or 5%, then we've seen the big effect of that. The second point, sorry, I want to ask also within the keto diet should be through the healthy food style or healthy foods. So the amount and the quality of the fats that we take should be very healthy and not the. And I'm sure if you have fed the mouse with the healthy fats, because I'm sure you just gave them trans fat, all these, hydrogenated fat, the results will be much, much better. Thank you very much.
Let me comment actually two interesting points. Ketogenic diets got a lot of traction in the press because it is an easy way to lose weight for people. It got a very bad press in the medical world because the initial proponent of the ketogenic diet was Dr. Atkins, who promoted eating bacon and sausage. And so promoted, substituting carbohydrates by highly saturated animal fats, which at the end might have actually not been helpful. There's a new version of the ketogenic diet, I totally agree with you, which is focused on plant-based, sort of much more healthy fat. I should say also, if I can share with you what our mice were eating, it was Crisco. I don't know, this is something that if you're in America, it's the most horrible, unnatural fat that you could possibly imagine. And the fact that these mice would live longer under these conditions to me is still really interesting. And I think there is still need to do an experiment with really highly much more healthy fat type of diet.
Experiments, you mentioned you have equivalent amounts of protein, 10%, but I don't know if you clarify, do you in this experiment also have total calories control, total calorie intake?
Yes.
Can I ask a question? So, Richard Veitch's ketone esters, as I recall, they're just two beta-hydroxybutyrate that are sort of fused together, right?
Yes. One actually had methyl groups on the hydroxyl.
Yeah. And so why is this more advantageous than
Yes.
this? Or why, and why is that?
Because so he was solving one out of the two problems. The first problem was absorption, getting rid of the salts. Ours actually provides another sort of think about it, a longest source of endogenous ketones.
Right.
That was really...
Okay.
And we've compared ours to, Richard Veitch's ester is actually also commercialized.
Right.
We've compared his to ours.
Yeah.
Ours gives a much longer, for reasons that we don't actually fully understand. Maybe because the hydrolysis is slower with a fatty acid versus just a methyl group. We don't know, but ours are clearly more delayed.
And one last question. It's injected?
No, it's oral.
It's oral?
Yeah.
Okay, all right.
You can buy it on Amazon if you want to try.
Fantastic. Amazon is not a sponsor. Just want to point that out. But they're welcome to sponsor. No. Okay, thank you so much. We will have a break. Fantastic talk.